Hello, ladies and gentlemen. Welcome to the lounge room where I'm going to now perform my. No, I won't. So I'd like to talk with you a little bit about observational research stuff. And I guess I only have 20 minutes, so I really want to try to impress upon you the importance of stepping back a little bit with our spheres of trying to conquer the diseases that we have with our investigational agents that we really think are going to work, and reassessing whether it is that we're ready to do that, and what stepping back and observing and performing some of these studies are. In talking with a lot of you guys, sometimes it would drill down to, well, what do we know about what actually happens normally? You know, are we ready to make that assumption on an effect for a given drug? Hopefully this will be interesting for you. So, you know, as you know, observational studies are kind of unsexy in the sense that we're not actually intervening. We're just stepping back and watching. And there are many different kinds of observational studies. But first, why would we do them? Well, I mean, there are cases, it's not so much in our cases, but in our cases, sometimes a randomized controlled trial would be unethical, like in an emergency type of situation, for example. One of the famous fun ones would be whether or not parachutes work. I think that there was a famous sort of study to determine a randomized controlled trial to see whether or not parachutes work. So in the study, they randomized people to an empty backpack versus a parachute and asked them to jump out of an airplane to see whether or not that would work. And you might think that you can't recruit for that study, but they did. This was the plane, and you can see a participant jumping out of the plane right now. This is a nice example of when you have a title for a study, or when you're making assumptions, and that's kind of one of the big points here, is that we make a lot of assumptions when we're designing our study, and we have our placebo and we have our investigational agent, but we don't necessarily know what placebo should be doing, and I think that's one of the reasons we do observational studies. So the other reason sometimes people will do an observational study is to try to see if there's a correlation or find an etiology of something, and then in the treatment of rare disease, observational studies can be very important in trying to know how to design that study as well. So sort of classically, there are different types of observational studies. We're gonna get to natural history studies, but observational studies, that we might consider in medicine would be case controls, people with the disease, people without the disease. And we want to try to match those groups as you know, just like you're doing in your interventional study. The nice thing about that is, and in clinical practice, we have sort of anecdotal evidence, but in talking to some of you about your protocols, a lot of times there's just dogma about what happens with, for example, the MRI in someone who has this or that kind of thing. And so there's a lot of speculation about what actually happens, rather than data that shows you what you could actually apply to the design of your interventional study. And so that's one of the things we were talking a little bit about earlier. The other thing is actually observing just within the cohort. So a lot of just sort of population studies that go on. This is more on the epidemiology side, but if you have a large group that has an exposure, for example, to pesticides or something else like that, and then you follow them over time, you might see that there are some people who get affected, some people who don't, and those types of things help you to try to suss out cause and effect. So those are studies that are of interest, but they are not necessarily as useful as a case control. And then probably the least sort of impactful would be a cross-sectional observational study, but and those are where we're really looking at a specific point in time and comparing people at that specific point in time for whatever thing that you're looking at, and that can help with prevalence, and genetic studies and things like that might fall into that category. But for us, we're going to be really looking at more of a natural history type of thing, which is can be either a cohort case control. So the bad thing, of course, about observational studies, and the reason that I think junior faculty and sort of early career trialists aren't that excited about it is because you're not actually doing anything to help the disease, and so you feel kind of helpless watching that. And then the other thing, of course, is confounding variables. I mean, when you're observing a cohort, there's just all these variables that you're not necessarily controlling for, and that can be quite confusing. I'm a big fan of the XCDY comic or whatever. I always get that right. It's my dyslexia. But anyway, I think that's pretty funny. But the nice thing about it is that in some cases, in particular for rare diseases, rare fatal diseases, it may not be as, you may just want to give something and see what happens, although that's not going to help with the FDA. Sometimes observational studies can just be cheaper and quicker. They are more easily designed, and you can study rare diseases there more easily. But let's talk about natural history. This is the kind of thing that I think, it is a type of either case control or cohort study, but depending on what it is that you want to do. But this is something that is an observational study where you are usually basing it on a diagnosis or a gene test or some other kind of biomarker, an MRI scan or something like that, that you need to know how that population behaves over time. And that's the missing piece. That's the piece that sometimes we just assume our placebo arm is going to pick up. Well, it's a placebo, so that's going to be the natural history. But the problem is that a placebo arm is a biased population of people who have actually enrolled into the study, and that is different than the entire population of people who are there. You can't just assume that you know. And furthermore, you don't know what the outcome measures are in the population that is receiving the placebo a priori, because you are not doing those outcome measures on those people. So what we do a lot is, of course, retrospective studies. That's a nice way to maybe get a sense of things in your disease space or in your population, and it is reliant upon the quality of the data that existed before. And I'll tell you, as someone who's is very critical of data, is that the clinical data is really weak in retrospective studies, because in general, unless there's registries and unless there's really an attention to acquiring data in a systematic way, the clinical literature is incomplete. And often, because it's being done by people who are seeing tons of patients all the time, it's not necessarily as accurate, reliable, or consistent as what you would have in a trial setting. And so it is not the same. When you look at retrospective data, you have to be very skeptical, about what it is. It gives you a sense of what's going on. So the superior design would be a prospective study, and this is what I'd like to seed. In 20 minutes, I have like kind of one seed to plant, and that seed is that you should consider. Before you race ahead to be the hero with your drug that you're sure is going to, make a big thing, and I want it to, and I hope it will, make sure that you actually have a very good understanding of the natural history, and perhaps design a prospective study that you can mimic in a way the interventional study to get those outcome measures before you start subjecting subjects to risk, and also it will help you immensely in designing the interventional study. Does that make sense to everybody as far as how that goes? We'll go into that in a sec as far as why that's not a placebo, but it is a comparator that you can use. Yeah.
So for a retrospective study,
yeah.
If you draw the data out of a registry, does that improve the quality of that?
Definitely, and that's why there's been a lot of effort from the NIH and the foundations and so forth in different disease communities to fund and help support the infrastructure for those registries. And so, when we have patients in ALS clinic or MDA clinic, we try to get them registered so that there is some data that is systematically captured with common data elements and all of that. I'd say that things are improving and are much better. But in talking with some of you, I mean, some of you are doing diseases where that A doesn't exist, or B, there's a question as to, where you would get that data. And that's one of the things that I think was a learning curve, for me in the past, is sort of, how are you going to access all of that data and reconcile the fact that some of these databases and registries by all these well-meaning great groups, they don't necessarily match with one another. And so you have to do a lot of work there. I think, the NIH and foundations are working hard to make that a lot better than it used to be.
Steve, can I say one thing?
Yeah.
This is an opinion.
Yes, I'd like to hear your opinion.
One of the problems with, this goes back to the thing yesterday, one of the problems with these natural history studies is they ask the clinical experts to set them up and don't bring data management experts in to set them up.
Yeah. Yeah, I mean, we're going to get to that part too. I think that's a great point. So, I think it was Chris who said this, but this is something that, I also always ask trainees, whether it's in the lab or in the clinic, and that is, so what? And to many of you, I've done that. Why would you do a natural history when I've got this thing that I know is going to work? And so it really is interesting, because I think by diagnosis in many of our diseases is heterogeneous. So, it's not, unless it's sort of a gene, but even a gene diagnosis, different variants can have different phenotypes that are going on there. So if you don't understand the complexity and the variability in your population, then you may have quote unquote responders, non-responders, that type of thing. That's what a natural history study would be helpful for. So it might help you identify within your diagnosis a population within that is of interest, particularly if you're doing these different outcome measures. You can validate some of the outcome measures that you want to do, like motor function tests and so forth, in a natural history study. And then, we're talking about biomarkers, but biomarkers are a great way to fold into a natural history study that you're prospectively designing, because then you're doing things that would not necessarily be a part of the normal clinical management, but things that would be a part of your clinical trial that are going on. Again, I think that a lot of times we just sort of throw biomarkers out there without strong justification as to why it is that we're using, X, Y, or Z. And a natural history study that is well designed can have your test of whether, for example, the muscle MRI or the ultrasound on those types of things, to give your example, whether, how do they actually behave in a prospective way in these patients. That's something that you wouldn't know from a retrospective study, because it's not being collected. And then, this thing about the non-concurrent comparator, what that means is, well, can't I just use that now in as my placebo? And the answer is no, you can't. And the reasons for that really are very interesting. I had the privilege of running an SMA natural history study through Neuronext with infants less than 2 years old. This was prior to the development of therapies. We knew that those therapies were in the pipeline. And what was interesting is that recruiting patients into a natural history study is different than recruiting into a treatment study. And so in the case of in an environment where there were no therapies, moms who donated their babies, volunteered their babies for this kind of study, they are not necessarily going to be the same as aggressive in their management of the infants as, say, someone who's very motivated to get them into a gene therapy study, for example. And so there are differences in their concerns and differences in how the management of those patients would be. So they're not by any means a placebo, but in some cases, and particularly very rare diseases, it may not be feasible to have that placebo. And are there any other comments? Robin, do you have any comments about that with respect to trials in which there's no placebo versus and using a non-concurrent arm?
My only comment is that the FDA generally likes a placebo
Right.
And another comment is that CDC is, for example, sponsoring the registry for ALS, not NIH. NINDS typically doesn't sponsor registries, but NCATS has as part of the Rare Disease Network.
Right. Yeah, for the case of ALS, it's got CDC there, that's right. And so speaking of the Food and Drug Administration, they have guidelines on how to do natural history studies with label and with approval in mind. And so I would look this up if you're interested. This is something, one of the two main points that sort of came out of this are to make sure to engage the patients, patient advocates, and caregivers into the discussion of outcome measures in this kind of natural history study. And also to make sure that through this process you're able to validate these different outcome measures that can be used in the study for regulatory purposes. So I think that there's not a lot to talk about these studies. Yeah, this is observational bias when people know they're being watched. But I think that the main things, again, that one seed is just to step back and really be critical about what you think you already know about your patient population and about what's been done. And if it doesn't exist, consider doing a natural history study or having a natural history study, in a way, having a lead-in, if you can, into the beginning of therapy so that you are able to have a successful study. So some of the keys to success that I have here, I don't know if this is a complete list really, but in order to get funded for a natural history study, which is a little bit more difficult because there's no sponsor who's trying to make money off of a drug, for example, or a device, is the timeliness of the need of the data, right? So the in the case of SMA, for example, the NIH, really wanted a biomarker natural history study because we all knew that these gene therapy studies were going to start. And so timeliness was really the big thing there, is that was data that every pharmaceutical company that was getting ready to do a gene therapy study really wanted. And I think what you will find in your different disease spaces is that industry is very, very motivated to move their product forward, and they want the academic to help them. But they also want the academic to provide them with targets and diseases and all that stuff. But the one thing that they don't tend to do a good job of is fund for that type of natural history type of thing. So this was, the Neuronext study that I was able to run was an excellent example of public funding that provided really a very valuable data set. Even though we didn't have any intervention in the study, that industry definitely used to accelerate their programs to the current therapies that are available. The other thing about a natural history study, when we talk about cohort study, but the other thing with a case control natural history study, which is what we did, we actually followed healthy infants exactly as we followed the SMA infants. And of course, the issue there, we did EMGs, we did blood draws, we did all of these things, and that data didn't exist. Because why would you do that? Why would you do that? So we didn't know what a normal CMAP is, in a 3-month-old. We didn't know, not in a controlled way, not in a way that we would go through the study. And so if you do a natural history study, in a way that you do it in a cohort and you have a control arm, actually, you can actually have the width of where your trial will fall inside. It's going to fall somewhere in that healthy range versus the untreated range. And that is very helpful for our biostatistician friends to know what the ceiling is, what the floor is, and these different kinds of things when you're going through. So, the other thing I've talked to some of you about is to try to just imagine the perfect experiment, rather than what it is that you can do, and then work from there. I already kind of said that, in an ideal situation, if you already know something is coming down the pike, consider designing a prospective natural history study that would be essentially designed for the most part, just like, or timed similarly to what you would do in the actual interventional study if possible. This is something it sounds like you did prior to getting ready to, for your gene therapy, which is outstanding. And so, and then the other thing, it really hasn't been talked about at this meeting or at this course yet so much, but I'm pretty passionate about this, is to think about your recruitment and retention plan early and often. This is true for your interventional studies, but it's particularly true in studies that are observational where you're not offering a drug. Why would someone volunteer for this? And so it is very important to kind of put together a program and think how it is that you're going to approach this community without being, sort of unethical about, false promises about what will happen if they do this. The fact is that, in the natural history study, where you have a fatal disease, for example, patients are going to die. And while they're getting ill, you really want them to continue to come in so that you can study them. It's a hard ask. And so you really have to do a lot, not just on recruitment, but on retention. So I think that, I don't know if there is a thing just on recruitment or retention, but I do think that is something that, is, I guess would be my second seed for you as you are, moving forward as young clinical trialists. So that's really all I had as far as, what I wanted to plant in 20 minutes for the seed. Does anyone have any questions about natural history studies or how to design them? Or Kristen, do you want to add anything to that?
The last point you said, I will add a point
Yeah.
that, so the microphone. Just the recruitment or retention, since we don't have that on here. This came up at our table last night. One of my soapbox moments, this is kind of a segue, Steve. Like with university grant support programs, is there lots of grant writing workshops you can go to that will teach you how to write a grant? There ain't a lot that teaches you how to be successful with your grant and work to get another grant. That's not unlike that. There's a lot of emphasis in clinical trials on recruitment, and are we recruiting on target? Are we recruiting on target? And there's actually less emphasis in retention, which is equally, if not more important. Like it doesn't matter if you hit your recruitment target if nobody stays in to the end, and if nobody that stays in does the things you wanted them to do in that. And so I think it's a similar analogy, right? Even though it's a slightly different thing, but it's not just meeting your target in recruitment, but it's keeping them in. And in these type studies, it's really important because as you get longer into natural history studies, missing data could very well be informatively missing data like we talked about yesterday, because the people who are doing worse stop coming in, and you don't account for that when you're using these data to plan trials. You're gonna have flawed designs in your trials.
Yeah, and I think that in talking to potential participants, it's just about being transparent and open and saying that the purpose of this study is seeing things over time. So, so if you aren't able, or you don't feel that that's something that you're going to be able to do as this disease process goes, then we don't want you in the study. We're still going to love you and manage you and be your doctor, but we don't want you to be in this study. And people generally are open to that kind of honesty, about what's going on. And from a cold-hearted data side, you don't want that person in your study. You just don't want them, going forward. I also recommend scripts for this, especially for multi-center studies, to ensure that the coordinators and the PIs and so forth are all speaking to the patients in the same language with the same messages, and that way, that might help with sort of site-to-site variability. Another thing that we see a lot, which we think is because of how people approach patients about research. So natural history studies can be difficult for recruitment and retention, so really want to think about it.
Let's give an example from your study that like little things can help keep people in, back on the retention thing. Steve, actually early on, I can't remember how it even came about, but there was a company in North Carolina, right, with the onesies. that they sold these onesies that were these blue onesies that said super baby with a yellow cape.
Yeah.
And so they actually convinced them to donate them for the study. And so every time a baby was enrolled, they got the super baby onesie. And Steve actually even had an adult super baby, not a onesie.
So this is all A.B. Bartlett, who was the superstar, and the reason that this retention plan was as successful as it was. I just did what she told me, but she's the one who.
Which was the right move.
I learned that a long time in my career. But I think that but yeah, she and her, she had a mom's group who had identified that website. And so, yeah, every baby got a onesie with a cape, and it was a theme. We had printed thank you cards. So there was actually like a real theme to the whole thing. And then I would go to the QRSMA meetings and other things like that, and I had I had the same thing with a cape. And I would, run and do that kind of stuff. And we had patient posters, at the QRSMA meeting, which many meetings have this sort of poster sessions for the parents. It made people, what it does is, people say, oh, well, there's no benefit to this. But there is a benefit to the offer of research, even when it's not interventional. There is a benefit, like in ALS clinic, people donate, tissue for us, knowing it's not going to help them. There is a feeling of being a part of a community. There's a feeling of being a part of an answer, even if it doesn't help them. It's one of the best parts of my job is hearing, working with these people who are at that place in their lives where, these are adults, of course, they're not kids. But I think that when you go after it, one of the things that people have really reported on as far as determinants of good recruitment and retention also is the attitude of the physician talking to the patients. If the physician doesn't think that this is, it feels badly because, I'm just gonna watch, I'm not gonna, then, there's not gonna be good recruitment and retention. You have to be fired up about why it is that this is important data. The timeliness is one of those things, but these parents who lost their babies who were in this study, they feel a benefit. They feel that they have contributed to the future that went on, and that is worth it. All right, Rudy. Oh, yeah, question.
Chris, earlier you mentioned something about data management, getting somebody involved early.
Yeah, so
On that?
yeah, my point was, so you need two things with the natural history study, right, to get a lot of the advantages that Steve mentioned. First, you gotta do it, but you also need to do it in a way that's gonna be useful. And so there's been too many times when I've worked with like a natural history database that was set up that wasn't set up with a lot of the things we talked about yesterday, right? So you can't tell missing data from null data, or there's, the data management 101, do not ever, nothing else data management, do not ever ask the same question twice on CRFs in the same study, because you won't get the same answer sometimes, and it's gonna drive you crazy, right? So things like that. So if you have it, it's almost more frustrating to have a data set that you can't use than to have no data set. You can still use it, but it may not be as useful for what you want to. So part of it in setting it up, it ideally, and I don't think we're really there yet, mostly because data management's not really a field you go into to learn. People kind of back into it from different areas. But in setting these up, and they're often not funded extremely well to get started because you're trying to get it going from the start or whatever. And I no offense to all of you clinicians in here, but the absolute worst people to design a case report form are clinicians. Because often they're set up like a medical note where you want to make sure you capture everything, lots of text fields, do not use text fields on your CRF, right? But there's an art to good data collection. And my point was often, if that's not part of this, you get some of the benefits that Steve talked about, but it's not going to be as useful as you really want it to be.
Yeah.
And then one clarification, so if you do a prospective natural history study, that would by definition be a registry study or not necessarily?
Not necessarily.
No, registries are, repositories of data, that people, in clinic, for example, are signing people up for. And the registries, depending on the registry has some kind of common data elements that's there. May or may not be good enough. My enthusiasm and my encouragement about a well-designed prospective study is that you can design it in a way that is the most useful for the oncoming clinical trials that are going to be going on there. And part of that includes, importantly, how you're going to report the data. Because you don't, with the clinical hat on, I don't, sort of record things in the chart in the same way that I would in a trial.
So a good example of that, I worked in PD with the Michael J. Fox Foundation, and they started PPMI, which is basically a large natural history observational database in 2011. But their goal in setting that up was not necessarily to do a natural history study for the sake of doing a natural history study. They were at the time trying to get better data to encourage pharmaceutical companies to do more clinical trials and develop more therapeutics in the area. And so it was very much what Steve mentioned. It was basically an observational study set up, but set up with a very clear goal of helping to grease the skids and provide information that would, in essence, de-risk pharmaceutical companies from, getting more into the PD therapeutics field.
All right, let's give Dr. Kolb a hand.